Greetings. I am AINA NADHEERA, your Chief Scientist in Prime Pharma. Okay, our agenda for the meeting today is to understand a strategy that I have came up for drug development for type 2 diabetes mellitus. Okay, but first let's get to know briefly about the disease. Okay, we will target Malaysia since the current survey in Malaysia in 2019 showed that 1 in 5 adults in Malaysia have diabetes. That means about 3.9 million people aged 18 years and above have diabetes. Okay, from this figure, you can obviously see the increasing trend of diabetes from 2011 to 2019. And MOH Malaysia has predicted that the prevalence will be continuously increased until 2025. Okay, so we will focus on T2DM because it is the most common type of diabetes with the central etiologies of insulin resistance and progressive deterioration of pancreatic beta cells. Okay, therefore, we as one of the renowned pharmaceutical companies need to produce at least one anti-diabetic agent to be distributed to the world, especially in the countries with high prevalence of diabetes like Malaysia. Okay, the problem with the current available anti-diabetic agents in the market are that they cannot reverse or inhibit the progression of T2DM and also have many severe side effects. Okay, also the process of drug discovery consumes a lot of money, time and resources. So our goal this time are to generate a novel drug that are effective for glycemic control, which can simultaneously regulate long-lasting blood glucose concentration while at the same time saving money and reducing the duration for the whole drug development process. Okay, moving on to the drug discovery. So I have decided to use computer-aided drug design, or CAD for short, as our strategy for drug discovery. Okay, starting with target identification, there are several possible targets for diabetes such as alpha amylase, glucosidase, G6P, captangene, and many more. Okay, so target identification will be done using bioinformatics, while for target validation, we will use target druggability prediction. Okay, next is hit discovery using ligand-based drug design. Okay, ligand-based drug design is an indirect approach where we will predict how the ligands bind to the receptor without knowing the structure of the receptor. There are many tools that use ligand-based drug design, which are shown in the slide. Okay, so ligand-based pharmacophore modeling is the most common tool to explore the molecules that trigger the desired biological effect. This can be done by creating a pharmacophore model, encoding the correct 3D organization for the required interaction pattern with the target of interest. Okay, next is 2D similarity searching, which is also one of the most applied approaches to identify the compounds with desired properties from several databases. Okay, other than that is QSAR modeling, or quantitative structure activity relationship. It is another computational modeling method to identify the relationships between the structural properties of chemical compounds and biological activities. Okay, this is one of the powerful methods as it provides high and fast throughput and good hit rate. Okay, last but not least is scaffold hopping. Okay, it is used to search for the compounds that are structurally diverse and share a particular biological activity by preserving the 3D interaction properties of a scaffold while changing the structural skeleton. Okay, double-headed arrows shown here indicate that these techniques can be used interactively in several iterative rounds of ligand design. Okay, next is lead discovery, which will be done by using virtual screening, which is the alternative of high throughput screening, HTS, mainly in terms of cost effectiveness and higher chances of finding most appropriate novel hit through filtering huge libraries of compounds in silico. The other approach is docking and scoring. Okay, lead discovery will then be followed by several cycles of lead optimization and subsequent lead discovery using LBCAT. Okay, the methods include for lead optimization will be scaffold hopping and 3D QSAR. Then for preclinical studies, we will conduct in silico ADMET prediction and physiologically based pharmacokinetic or PBPK simulations, which will be integrated into in silico lead discovery stage to reduce the cost. Then the selected compounds will be the drug candidates for further studies in drug development. Okay. This is the example of the study that used ligand-based pharmacophore modeling, virtual screening, and molecular docking to find novel compounds for diabetes. Okay, so 3D structure of drugs was performed using PubChem. 3D structure of the targeted gene was conducted using PDB. Set compounds as test and training. Generation of shared features pharmacophore and screening with libraries were performed using Ligand Scout. And then to estimate the drug's likeliness property, compounds that fulfilled the Lipinski rules were selected from zinc libraries and their toxicities were assessed using Protox server. And then the downloaded hit compounds were then docked for validation using PatchDoc server, and they were compared and analyzed using BioVia Discovery Studio to identify the distance and types of bonds between the receptor protein and ligands. The compounds with strong binding interaction of ligand and the gene of interest will be selected for further tests. Okay, last but not least is the basic timeline overview of drug development. Okay, so after drug discovery and preclinical studies, we will proceed with drug development phase using selected drug candidates for clinical trials consisting of 3 phases to see the safety and efficacy of drug candidates. Okay, next is to get FDA review and approval. Only after the drugs are approved, then we will release them to the market. Okay, lastly is the post-market surveillance to see the long-term effectiveness and its side effects. Okay, in conclusion, since CAT is a very cost-effective, time-saving, and mostly using rapid and automatic process in comparison with traditional HTS, hopefully we can find the most promising drug candidate effective for T2DM with minimum side effects and high success rates using less than $1 billion within 15 years. Okay, these are my references, and that's all from me. Thank you.